For the estimated 55 million people worldwide living with Alzheimer's disease, the reality is a slow, terrifying erasure. Memories, personality, and identity fading into darkness as the physical connections in the brain simply disappear. For decades, the medical world rallied around one foundational theory to stop this: the amyloid hypothesis. The idea is that Alzheimer's is driven by a specific protein that misfolds into dark, sticky clusters, amyloid plaques, that build up on the outside of brain cells, choking off their ability to function. Following this logic, the pharmaceutical industry spent billions of dollars and countless years hunting for a single chemical solution. The goal was straightforward: invent a drug that could target and flush those plaques out of the brain, and you cure the disease. That brings us to a massive recent review from the Cochrane database.
Researchers pooled data from 17 different clinical trials, encompassing more than 20,000 older adults who had taken these anti-amyloid medications over many years. This chart shows the aggregate results of those trials, and the conclusion is stark. Over an 18-month period, the data shows a completely flat line for cognitive improvement, meaning the drugs provided almost no meaningful relief for memory decline. Even worse, the red line below shows a sharply rising risk of severe physical side effects, specifically swelling and bleeding in the brain. This creates a terrifying vacuum in the medical community. If successfully clearing the plaques doesn't actually stop the dementia, the foundational premise that has driven modern Alzheimer's research for a generation might be pointing us in the wrong direction. But clinical neurologists are pushing back hard against that broad, pessimistic headline. To understand why, we have to look closely at how this specific study was constructed. This chart shows the statistical trap of pooled data. The researchers averaged the results from a wide timeline of trials. The massive cluster of gray data points represents older drugs that completely failed. By lumping everything together, those heavy failures statistically bury the tiny cluster of colored points, representing the newer, FDA-approved therapies. If we remove the noise and isolate the newer drugs, like lecanemab and donanemab, the picture changes. These specific medications actually succeeded in crossing the blood-brain barrier and successfully clearing the amyloid plaques. However, the reality of that success is incredibly modest. When you test patients taking these successful drugs, you see a statistically significant slowing of cognitive decline. But in practical terms, that slowing amounts to roughly a 1 or 2 point difference on a 90-point cognitive scale. This leaves doctors and families with a difficult calculation. You have to weigh a tiny retention of daily memory against a frequent, documented risk of brain swelling. What this tiny statistical win reveals is a crucial biological reality. Clearing amyloid is not a failure, but amyloid alone is only one driver in a network of intersecting biological failures. To find more effective targets, researchers are expanding their focus to other mechanisms breaking down inside the brain. The next major target is tau. While we know amyloid builds up on the outside of brain cells, tau causes problems on the inside. Toxic tau proteins form tight tangles right inside the cell core, directly choking off its internal pathways and causing memory loss. A second alternative target is neuroinflammation. Scientists are looking at how the brain's own immune system might become overactive, turning a protective response into chronic inflammation that accelerates the cellular damage. Then there is the heart-brain connection. We now know that managing a patient's vascular health, blood pressure, and energy supply is critical. If the blood vessels are damaged, the brain loses the infrastructure it needs to protect and repair itself. Alzheimer's attacks the brain through amyloid plaques, tau tangles, runaway inflammation, and vascular failure all at once. Because it involves so many distinct systems, a single drug targeting a single protein is unlikely to stop the disease entirely. The strategy for Alzheimer's is shifting toward the model used for complex diseases like HIV and cancer. Treating a highly resilient illness requires combination therapy, attacking the condition from multiple angles using a precise cocktail of different drugs simultaneously. In the future, a clinical visit for Alzheimer's will be highly personalized. A doctor will look at a patient profile measuring their specific amyloid load, tau density, and vascular risk, and then prescribe a custom matrix of treatments built uniquely for their biology. When you view the science through that lens, the massive Cochrane review of failed medications isn't a dead end. It is a vital scientific self-correction. By understanding exactly why these early drugs failed across tens of thousands of patients, researchers are learning how to refine their targets. Those failures teach us how to select the right patients for the right therapies. The search for a single miracle pill has been replaced by a focus on precise, multi-targeted interventions designed to protect the brain from every angle. It is our best shot at keeping the lights on.